DNA Replication by Alex Mazza and Joe Schmidt. Step 1. Hydrogen bonds between the bases of the two strands. Where the two strands unwind is the starting point. Splitting happens in places of the chain which are rich in adenine and thymine. This is because there is only two bonds between them, as there is three in cytosine and guanine. Helicase is the enzyme that splits the two strands, breaking the hydrogen bonds. This breaking occurs because the enzymes lower the energy needed. Step 2. During this step, the RNA primates bind. RNA primates attract RNA nucleotide which bind to the DNA nucleotide due to the hydrogen bonds between the bases. RNA nucleotides are the primers for the binding of the DNA nucleotides. Step 3. Makes the two strands in different directions. In one of the strands, the ligase comes through and connects them. Step 4. Exonuclease. The fragments remove the RNA primers. The gaps are closed with the actions of DNA primers. Adds the phosphate in the remaining gaps of the phosphate sugar backbone. Each new double helix is consistent of one old and one new chain. This is what we call semi-conservative replication. Step 5. The last step in termination. The process happens when the DNA polymer reaches to an end of the strand. The end of the parental strand where the last primer binds isn't replication. These ends of linear DNA consist of non-coding DNA that contains repeat sequences and are called telomeres. Telomeres are a region of repetitive nucleotide sequences at the end of a chromosome, which protects the end of the chromosome from deteriorating. As a result, a part of the telomere is removed in every cycle of DNA replication. Step 6. It is not over until mechanism of repairs fixes possible errors caused during the replication. Enzymes remove the wrong nucleotide and the DNA polymerase fill the gaps.